Hi, I'm Danny from Taiwan. I would like to thank the organizers for the opportunity to share our recent structural studies on the SARS-CoV-2 spike variants by cryo-EM. The past 2 years have witnessed multiple waves of COVID-19 infections, leading to millions of confirmed cases and millions of deaths. Multiple variants of concerns or variants of interest have been reported. They have different mutations in their genome, particularly in their spike proteins. SARS-CoV-2 used the spike protein to recognize host cell receptor ACE2. It is important to note that both the spike protein and ACE2 are highly glycosylated as indicated in the color sticks on the right. These post-translationally modified glycan structures provide camouflage to help the virus evade from host immunity attack. So today I would like to tell you how we combine cryo-EM and mass spectrometry to investigate the structures of these spike protein variants, particularly in the context of glycosylation. I would like to also discuss how these mutations affect the structure-function relationships of the spike and how we can develop better antibodies against COVID-19. Our cryo-EM facility was established at the end of 2017. At the time, I collaborated with my colleague at National Taiwan University to study the structure of a cat coronavirus known as FIPV. FIPV infection is very lethal for infected kittens, and so far there is no effective treatment or vaccines available for FIPV. Using recombinant FIPV spike protein, we demonstrated the ability of mass spectrometry to determine site-specific glycoforms of individual glycans, and surprisingly, we are able to define for many of the glycans high-resolution cryo-EM maps for the individual glycans. As illustrated in this animation, it is essential to make the best of cryo-EM and mass spectrometry. Cryo-EM provides the EM densities to help us locate the glycans, but it requires the chemical information derived from mass spectrometry to help us build the atomic models. In the case of SARS-CoV-2, however, the glycans are often too dynamic to be defined with clarity. So we often had to resort to unsharpened EM map that compromises the EM resolution, but it is able to help us locate the spatial distribution of the individual glycans on the EM map of the spike protein, as illustrated in green in this unsharpened EM map. As the pandemic progressed, multiple variants of concern or variant of interest have been reported, and all these variants share the common mutation that is the D614G mutation. Shortly after the first report in China, the D614G mutation was reported to dominate the global infection cases. We purified the recombinant spike protein of both the wild-type D614 as well as the mutant harboring the D614G mutation. As illustrated on the left, the wild-type recombinant protein started to lose its compact structure after incubation at 4 degrees for as short as 24 hours. After 6 days of incubation, the wild-type recombinant protein almost lost completely its native compact structure, whereas the D614G mutation retained the compact structure under the same condition. We next systematically checked the temperature-dependent denaturation of the spike protein variants of the wild type on the upper panel and the D614G on the lower panel. It is clear that the wild type is very sensitive to both cold denaturation and heat stress, whereas the D614G mutation seems to be very robust under the same condition. So we propose that the D614G mutation enhances viral fitness by desensitizing it to temperature-dependent denaturation. Further quantitation of the negative SEN EM images shows that in the case of wild type labeled as D, it almost completely lost its native compact structure at 4 degrees after 6 days. In the case of D614G, however, most of the protein remain native-like. Further differential scanning calorimetry analysis shows that incubation of both variant at 37 degrees Celsius for 6 days seems to retain the integrity of the spike protein as much as the freshly prepared samples. In contrast, for the wild type, incubation at 4 degrees Celsius for 6 days lost the high temperature transition peak as indicated by the dashed blue line. In contrast, the D614G mutation retained most of its high temperature transition peak. So the take-home message is that the wild-type protein is very sensitive to cold denaturation, and even a short period of cold storage is bad for the wild-type protein. We next used cryo-EM to determine high-resolution structures of the D614G variants. Using 3D variability analysis, we can derive 5 distinct structures for this variant, 3 of which correspond to 1 RBD up, and 2 of which correspond to 2 RBD up. In this case, we did not observe any ORBD down conformation as was observed for the wild type. Detailed analysis indicates that wild type protein forms an interprotomer cell bridge between D614 and Ka54 in the other protomer, as highlighted in the blue circle. Due to the mutation, this cell bridge was lost. Consequently, the RBD seems to lose the constraint and populating more in the upward conformation. We next investigate the structure-function relationship of the alpha variant that was first reported in the United Kingdom. Using the same strategy, the alpha variant, also known as B117, exhibited 3 distinct 1 RBD up conformation and 1 2 RBD up conformation. Further analysis to compare the RBD up and down conformation shows that the alpha variant specific mutation A570D is involved in an interprotomer cell bridge that compensates the loss of cell bridge due to the D614G mutation. Switching from the RBD up to the RBD down conformation requires the breakage of the interprotomer cell bridge involving A570D, and to form a new cell bridge again with A570D, but with a different lysine residue as indicated at the bottom. So we propose that this alpha variant specific mutation A570D serve as a molecular switch to regulate the up-down conformational change of the RBD in a pedal beam-like mechanism. Further structural analysis indicates that the alpha variant forms a new pi-pi interaction between the RBD and the ACE2, and this pi-pi stacking likely contribute to the enhanced binding affinity between the spike and the ACE2 as indicated by the biolayer interferometry kinetic analysis shown on the left. We collaborated with our colleague at Academia Sinica to solve a number of cryo-EM structures of monoclonal antibody in complex with the intact spike protein and define 3 distinct structural epitopes for antibodies 15, 25, and 45. For antibody 25, its epitope overlaps with the mutation site N501Y, and indeed, in the ACE2 competition binding analysis by BLI, antibody number 25 lost its ability to compete with AC2, as indicated in the dark blue trace in the middle panel. Because antibodies 15 and 45 have distinct structural epitopes that are quite far apart on the RBD, we demonstrate the possibility to combine these 2 antibodies as an antibody cocktail to form a 3-2-3-2-3 complex with the spike. The structure explains why these 2 antibodies are not affected by the N501Y mutation and how these antibodies effectively compete with ACE2 binding. The biophysical analysis was further confirmed by pseudovirus assay and animal model studies using mice and hamster. We next look at the effects of mutations on the other variants of concerns, and we use the same strategy to show that beta, gamma, delta, and kappa variants can sample a broad range of conformations ranging from ORBT down to ORBT up. In particular, we solved the ACE2 bound form structure for all the variants, and in this case, we derive an excellent correlation between the interface area with respect to the free energy of binding derived from the dissociation constant based on the BOI analysis, as indicated in panel D. Looking at the structures of the RBD, we also demonstrated how these mutations rearrange the surface electrostatic potential. That's very important to recruit AC2 binding. In addition, we show by cryo-EM that the hyperenergetic antigenic N-terminal domain supersite is remodeled due to the loop deletion and other mutations in the Delta variant compared to the wild type. The supersites are indicated in white or light green. Furthermore, in the context of glycosylation, we show by cryo-EM as well as mass spectrometry that the gamma variant introduced two new N-glycans at position 20 and 1A8. These two new N-glycans shown in pink will sterically inhibit monoclonal antibody to binding to the N-terminal domain, as previously demonstrated by other structural studies shown in panels E and F. Furthermore, the Delta variant has a T19R mutation that removes the N-glycosylation sequence for residue position 17 because of the requirement of NXS or T. So we believe this additional effect of changing the glycosylation pattern is important for the immunity escape for the Delta variant. Similar strategies were applied to all the variants, combining the BOI competition assay, negative stand structural determination for the antibody cocktail, and the pseudovirus assay. We demonstrated that our antibodies can still effectively neutralize different variants so far. So with that, I would like to summarize by saying that cryo-EM is a very powerful tool to provide detailed molecular insight into the structural function relationships of these spike variants. But in some cases, glycosylation plays an integral part to the functional contributions, but they are too dynamic to be visualized by cryo-EM. In this case, mass spectrometry is a highly complementary tool to help us derive further information. With that, I would like to thank my colleagues and the core facility in Academia Sinica to provide technical support for this work. Thank you.